So as you might know, I'm based at the University of Oxford at the Chinese Academy of Medical Science Oxford Institute, but I'm also part of the Center for Medicine Discovery. And today I'm gonna, well, first of all, give a brief introduction of my research background, and then I'm gonna explain a bit why we work on the ubiquitinating enzymes, and we think we are therapeutic targets of interest. I will explain the techniques we use to study DAPs. We use a lot of mass spec based workflows, so we call them Ubiquitomics, and I'm gonna explain a bit in a couple of them. In particular, I'm gonna talk about activity-based protein profiling, how we can use this for that inhibitor screen and characterization, and I will show you some examples we have identified recently with this platform. And then if we have time, I can discuss other area of interest in my lab, which is the acylating enzymes, in particular USP18, which is a target in immuno-oncology. So first of all, a bit of my research background is mostly cancer biology and ubiquitin therapeutics. So I did my PhD in Santander University of Cantabria, and there I studied the oncogenic RAS biology in the laboratory of Professor Piero Crespo. And we started to have an interest in drug discovery, and we were interested in identify drugs that would interfere with the resistance mechanism observed in the clinics with conventional BRAF and MEK inhibitors. And we identified a molecule that was disrupting a protein-protein interaction in this pathway that was overcoming resistance to these conventional inhibitors. Then I moved to UCL Louvain in Brussels, where I did a tumor microenvironment pharmacology postdoctoral stay there for almost 5 years, where I studied tumor hypoxia and acidosis. And we were also investigating ways of identifying drugs targeting specifically hypoxic tumors. So these are tumors that they don't receive enough oxygen supply because, as you might know, tumors grow in a not very controlled manner, and sometimes they have areas where the blood vessels don't reach, and therefore they become hypoxic. So one strategy to shrink tumors is to target hypoxic regions, and in that, during my postdoc, we identified a molecule that was able to target specifically hypoxic tumors, especially an interesting aspect of this drug was activated by light. So the combination of hypoxia with light activation, something that is called photodynamic therapy, will lead to a strong anti-tumor effect. And then I moved to Oxford to the laboratory of Professor Benedict Kessler when I did my second postdoc. And here in Oxford, I developed my interest in the ubiquitin system. In my previous postdoc, I started working on the ubiquitin system because these drugs will induce a strong accumulation of ubiquitinated material. And so that's how I met Professor Kessler, and I was very interested in activity protein profiling that I'm gonna explain a bit later. So that's we use these ubiquitomics techniques to characterize DAP inhibitors in the context of cancer. And here is where I work with a company called Forma Therapeutics, and we work in the development of a number of DAP inhibitors. They start always with the FT Forma Therapeutics. So we characterize USP9X inhibitors, USP28 inhibitors that we use in the context of squamous cell carcinoma in cancer. And then we, in particularly interesting for this foundation, we characterize very potent and selective USP7 inhibitors that I'm going to show a bit later as well, and USP30 inhibitors. these USP30 inhibitors are relevant for Parkinson's disease because regulate mitophagy is a Parkin-regulated process and is particularly interested in Parkinson's. And then at the end of 2021, I set up my own lab with the interest of expanding on cancer immuno-oncology. In particular, I was interested in studying the ubiquitinating enzyme regulating innate immunity. And my favorite lab of interest in this context is USP18, that if we have sometimes I'm going to talk about it, but I also was involved in discovery of additional molecules targeting not only USP18, but other the ubiquitinating enzymes like UCHL1, USP47 and 7 dual inhibitors, OTUD7B, and USP5 inhibitors. So across my research career, I was always involved in collaboration with industry. So in Brussels was a consortium with involved chemists and eventually led to a spin-out called Emerald Therapeutics. With Benedict's lab, I work with Forma, BMS, Bayer, Insight, and Pfizer. In my own lab, I work with Pfizer, and now I'm working with Boehringer and Ono Pharma. So why we are interested in the ubiquitinating enzymes? I think most of you already know what the ubiquitinating enzymes are, but just in case, so the ubiquitinating enzymes, they remove a post-translational modification of proteins. So the proteins can be modified with another protein, which is called ubiquitin. And this happens through an enzymatic cascade involving E1, E2, and E3 enzymes. So the ubiquitinating enzymes are proteins that remove this modification. They reverse the modification. And modification of cellular proteins with ubiquitin can lead to degradation of the protein or modulation of the protein. So you have an enzyme, metabolic enzyme, like in this case, ubiquitination can regulate the rate or even can inhibit completely the enzyme, and therefore the metabolite cannot be processed. So the message is that the ubiquitinating enzyme regulate the fate of proteins, cellular proteins that are more modified with ubiquitin. So it's not strange that the ubiquitinating enzymes have been found to be linked in disease. And here you can see an overview of USPs, which are cysteine proteases. You can see here a list of some examples of the ubiquitinating enzymes that have been linked to inflammation, cancer, viral infection, etc. And interestingly, inhibitors have been developed for many of these enzymes. In fact, some of them, they have gone into clinical trials. For example, we have USP30 inhibitor from Mission Therapeutic that is now being tested for heart failure. And also from the same company, they have a different inhibitor that was used for, that started to be being tested in Parkinson's. Also, USP2825 are being tested in AML, acute myeloid leukemia, and USP1 inhibitor, a couple of different companies are testing this in solid tumors, USP1 inhibitors. So how we study the ubiquitinating enzymes in the lab? So since we are a lab with a very strong proteomics background, so we have dedicated workflows to study ubiquitinated proteins. And one of the most popular is something called glycolide peptidomics. And I'm not going to go too deep in this, but proteins that are modified with ubiquitin, and when we trypsinize them, so this trypsinase is a way we use to cut the protein in smaller fragments called peptides. So they will be, it will be possible to analyze them by mass spectrometry. If you don't do that, it will be much more challenging to analyze proteins by mass spectrometry. So we digest them into smaller fragments, and the interesting thing here is when you have ubiquitin conjugated to a protein, it will leave two glycine bound to a lysine residue. So this is a unique motif that is only happening in peptides that were ubiquitinated. And there are specific antibodies that identify this modification and allow us to enrich for peptides that they come from proteins that were ubiquitinated. And this allows us to analyze by mass spectrometry which proteins are ubiquitinated. So for example, you want to know which proteins are ubiquitinated when you inhibit or make a knockout where you deplete USP7 in a cell line. You can identify which proteins become to accumulate ubiquitin, and this in theory is depending on USP7. And this allows us to identify substrates. We use this for several targets. So we use for the ubiquitinating enzymes, but we also use it for E3 ligases and E2 conjugating enzymes. So one of the cases that I normally show is USP18 depending IgG long. So we use this technique to study the substrates of USP18 when we knock out USP18. So we deplete USP18 from cells and we treat the cells with interferon. We can see a strong accumulation of IgG-latent material. So IgG15 is a ubiquitin-like protein. So it's a protein that modifies other proteins, but in this case is produced in the context of innate immunity. So when you treat cells with interferon, with this an antiviral cytokine produced in response to viral infection, you induce IgG15 and the conjugation of IgG-related proteins. And we use this technique to identify the substrates of USP18. We could identify substrates several substrates for USP18, and this allows us to understand what USP18 was doing in cancer cells. So one of the roles was regulating innate immune responses, and USP18 was modulating the IgG-lation of several double-stranded RNA binding and processing enzymes, and was regulating the machinery involved in antigen processing and presentation, and other pathways including immunoproteasome and chemokine-mediated T-cell recruitment. So all these substrates suggested that USP18 might have a role in how immunogenic cancer cells are. So this means how a cancer cell, when you deplete USP18, might be more visible for the immune system and therefore will be killed more efficiently. And that was the case. So when we did in vivo experiments, we could see that the immunodeficient mice, when we inoculate tumors of wild-type colorectal cancer, they grow tumors. And when we inoculate these tumors that they are deficient in USP18, here in green, we can see a smaller tumor. But the striking thing was when we did immunocompetent mice models, they have fully efficient immune system. The tumors, wild-type tumors, they grow slower than in the immunodeficient model. But when you knock out USP18, you have a complete remission of the tumor. So this means that the immune system is very important to eradicate USP18 deficient tumors. And this is in line with with the substrate we identified by using this ubiquitomics technique. So for example, one could think of a potential glypeptidomics experiment where we interrogate substrates in relevant models of Hauff-Fontaine syndrome. And this will understand better what these mutations of USP7, how they are affecting substrate recognition or ubiquitination of substrate of USP7. Another technique we use in the lab is activity-based protein profiling, and this involves activity-based probes. So these probes, I'm going to explain later, but they bind irreversibly to the ubiquitinating enzymes that are active. So that allows us to label the enzyme, but also allows us to isolate the enzyme with the probe. We also use structural ubiquitomics, which is based on also in a mass spectrometry technique. And this is based in the exchange of hydrogen by deuterium when you incubate with deuterated water proteins. And the interesting thing here is you have a molecule bound to the ubiquitinating enzyme. In this case, you will protect the exchange of hydrogen for deuterium. And this allows us to identify by mass spec these protected areas in the protein, regions in the protein. And this is very useful to characterize inhibitors, especially when you have proteins that they don't crystallize very well. So this doesn't require crystallization, but will still inform in the structure and dynamics of the protein. And there are other techniques more complex that allow to study topology of ubiquitin chains. As I don't know if you are familiar with that, but ubiquitin can be conjugated as a monomer to other proteins, but can also be conjugated as a chain of ubiquitin. So there are techniques that have been developed and use mass spectrometry to study the topology of these chains. Going back to activity-based protein profile and activity-based probes, we have developed activity-based probe for the ubiquitinating enzymes that they have the following architecture. So they have an specificity motif, which is ubiquitin itself. So this will allow us to bind only the ubiquitinating enzymes. They have the trap, which is a chemical warhead that will allow us to react with the catalytic reactive group in the ubiquitinating enzyme in an irreversible way. So they will create a complex between the probe and the enzyme, in this case the DAB. And it has also a visualization motif that can be a peptide or a biotin. So this is used to visualize the probe complex with the DAB or to isolate the probe complex with the DAB. And just, we don't know to go too deep here, but there are different types of warhead, and this will affect the reactivity of the probe with the different ubiquitinating enzymes. So why we are interested in this technique and in this ubiquitin-based, activity-based probes, because they can be used for structural biology studies. Sometimes, especially in the past, the proteins were challenging to crystallize, and they use DAB ubiquitin complexes to stabilize the complex and therefore to generate crystal structures. And these complexes involve these ubiquitin activity-based probes. So these probes can be used also to profile DAB inhibitors because they will compete. The inhibitor reacting with the catalytic system will compete with the probe, and that we will be able to detect that. And it will allow us to also study selectivity. For example, if you have an inhibitor, we can look at the probe and see whether the competition is global against all the DABs in the cell or just a very specific one. So one of the things we could use this technique, and we have conversation with some of the members here, is that it can be used to study the activity of the different USP7 mutants found in Howe-Fontaine syndrome. And it's also allow us to identify new ubiquitinating enzymes or IgG-lating enzymes in our case, and also interactors when we do combine this with mass spec. So how can we look at the complex between the ubiquitinating enzyme and the probe? So if we do a Western blot, so we allow the proteins to migrate in a gel. So you have here an example. And here a simplified example, the ubiquitinating enzyme will run at the expected molecular weight. However, if we have a reaction between the ubiquitinating enzyme and the probe, we will see a shift in the molecular weight corresponding to the probe. In this case, ubiquitin is around 10 kilodalton. So we'll see a shift with the probe of around 10 kilodalton. And if you have inhibitors competing with the probe, targeting the catalytic system of the DAB, you will have a competition, and the ubiquitinating enzyme will not bind the probe anymore. The other option is to isolate the complex using, in this case, an antibody against the probe that allows us to isolate the complex between the probe and the DABs, and we can analyze those complexes by mass spectrometry. So here you have an example for one of these USP7 inhibitors we characterized in the past. You can see here that USP7 without the probe runs at the expected molecular weight. I don't remember exactly, but it's around 130 or something along those lines. With the probe, we have a 10 kilodalton shift, meaning that USP7 plus the probe is detected by Western blot. And here you can see increasing concentration of this inhibitor is able to prevent binding to the probe. And in this case, was very potent inhibitor in the nanomolar range. And one interesting feature is when you look at all the DABs that have been labeled with this activity-based probe in increasing concentration of the inhibitor, you can see that only a band corresponding to USP7 can be seen and inhibited in this gel. So this means that these inhibitors are potent and very likely very selective against USP7. But by Western blot, you can see only a number of DABs, and you are not 100% sure that those are the ubiquitinating enzymes. So what we do is isolate the complexes, probe, DAB, and analyze by mass spec. So in this case, you can see here that two different concentrations of USP7 inhibitor was able to inhibit selectively USP7 and not other cellular identified DABs by mass spectrometry. So we were able to see different ubiquitinating enzymes in the cell. At the same time, they were profiled with this USP7 inhibitor, and we could confirm dose-dependent and selective USP7 inhibitor. So we applied this technique to other inhibitors in addition of the USP7 inhibitor. We had the non-covalent and covalent ones, but we also had the, as I said before, USP28, 25 inhibitors, also very potent and selective for 28 and also 25, because both are very similar structurally, and the inhibitors normally cross-react. USP28 inhibitors normally cross-react with USP25. We also could demonstrate selective and potent inhibition of USP9 inhibitor, and two different USP30 inhibitors we were also able to profile using this technique. So as you can imagine, this is quite powerful for drug discovery because it allows us to evaluate selectivity and potency in cellular environment. And if you have inhibitors as selective as these ones, this will very likely have less side effects when you treat patients with these inhibitors. So, but one of the things we found when we work with these assays is that they are limited by selectivity. So as you can see here, we can identify, and also you can see here very well that in the beginning we were able to identify between 15, 20 DABs. A few years ago, 30 DABs was a good number, but now we are over 40, 50 DABs. So one limitation is that we weren't able to profile all of them, so just a fraction of them. And another limitation is that these experiments had low throughput capabilities, so therefore were not suitable for drug screening. However, we were working on this over the last 9 years. So one of the things we work first is in the increased depth of this activity-based protein profiling, and we implemented high pH prefractionation in combination with mass spectrometry. And this way, instead of detecting 30 ubiquitinating enzymes, we detected 74 ubiquitinating enzymes. And we cover all the active DAPs that were supposed to be active in a particular cancer cell line. We had RNA-seq data, so we can compare RNA-seq data, deep proteome data with activity-based protein profiling, and we could confirm that we were able to cover all the active ubiquitinating enzymes in that cancer cell line. And in the recent years, we were making these assays higher throughput. So we implemented robots doing liquid handling and sample preparation to increase throughput. And we increase from traditional activity-based protein profiling, we increased the throughput 10 times at least by implementing these liquid handling robots and shorter gradients in the mass spec. And since when you increase throughput in proteomics, you normally lose sensitivity, we were also working in the data acquisition methods to make these workflows more sensitive. So now we have a workflow that is equally sensitive as the traditional LPPP, but with a higher throughput of at least 10 times fold increase. So the question we had, okay, we now made these assays high throughput. Can we make use them for DAB inhibitor screening? So we, as a proof of concept, we team up with a chemistry lab here in Oxford, led by Professor Chris Scofield, and we generated a compound library that was curated for potential molecules target the cysteines, which is the amino acid, essential amino acid for the activity of the ubiquitinating enzymes. And those include alpha, beta, and alpha deoxy ketones, DEX, gamma lactams, and also ubiquitin-derived covalent peptide inhibitors that were especially designed for this project. And we tested a selection of these compounds. So we tested 42 compounds in duplicates at a single concentration of 25 micromolar, and we use prefer cancer cell line for this screen. So this was the strategy, use the activity-based protein profiling as a screening platform, and also to validate the hit and select the hits. And then we did some compound design development, compound binding characterization using a structural proteomics that I previously talked about it. And then we did some functional validation to prove that the biology was aligning with the previously described data. So these are the results of the screening. So we identified 6 inhibitors out of these 42, and we could identify USP47 inhibitor, which was compound 3. And then we identified UT7B inhibitor, USP5 inhibitor, another USP47 inhibitor, and 2 USP33 inhibitors. Unfortunately, we couldn't validate these 2 inhibitors further, and this one resulted in a cross-linker molecule that was inducing protein aggregation and potentially will be very inspecific. Interestingly, the USP5 inhibitor called compound 25 was identified, and another isomer of this molecule that have this group pointing backward in this molecule was shown to be inactive. So this tells us that this platform is able to resolve isomer-specific effects, which is quite interesting in drug discovery. And also we can have now use this molecule as a negative control for further experiments where we use compound 25. Just to make a confirm being more confident on these hits, we use immunoblotting with the activity-based probe, and we could see that the USP compound 3 was inhibiting USP7. As you can see here, there is a competition with the ABPP probe, and also 47 seems to be more potent for 47 compared to 7. Compound 6 was inhibiting OTD7B, and compound 25 was also inhibiting USP5. And we could calculate IC50s by immunoblotting. And also we did the same, since this workflow, high throughput workflow allowed to run several samples, we test each inhibitor at different concentrations. And as you can see here for compound 3, mainly cross-react with USP47 and 7. Compound 6 inhibits UTD7B, but it seems to inhibit other enzymes at a higher concentration. And very interestingly, compound 25 only targets USP25 even at a very high concentration of 250 micromolar. Then we tested this drug in cells. So all these previous experiments were performed in cell lysates, but now we treated the cells with these drugs, and we confirmed that there was inhibition of the DAPs when we treated cells. So we could see this for 47 and 7 with compound 3, for OTUD7B for compound 6. However, we couldn't see inhibition of USP5 with compound 25. So this told us that this compound was either not permeable or not stable in the media. So the chemists generated analogs of this compound, and they improved permeability. And we identified 2 of these derivatives that were permeable and potent against USP5, while returning selectivity against other ubiquitinating enzymes. So the identified inhibitors, they might not be inhibitors that you will use in the clinics, but they were used as research tools. For example, we used compound 3, which is the USP40 inhibitor, which is a first-in-class USP47 inhibitor. Most USP47 inhibitors described were dual inhibitors with USP7, as they are quite similar in terms of structure. And we could confirm with recombinant protein that this compound is around 10 times more potent against 47 than 7, also in recombinant protein biochemical assays. We could see that in these biochemical assays, time-dependent experiments indicated covalent or low type binding inhibition of USP47, while USP7 inhibitor inhibition was weaker, as you can see here when you compare the curves here, and it was time-independent. And for both enzymes, the progress curves in the presence of compound 3 were non-linear, indicating a slow initial binding. And then when we did some modeling with the help of Andrew Turnbull, that was part of the USP7 study we did with Forma Therapeutics, he located the binding of the compound 3 in the cleft between the palm, thumb in the USP47. And so the ubiquitinating have this characteristic structure where you have like a hand shape. You have the palm, the thumb, and the fingers. And this compound seems to bind in the cleft between the thumb and the palm. And the modeling also identified interaction that is specific for 47 and it goes to the arginine 402. They will have an interaction with the compound 3. And this interaction cannot happen in USP7 because it doesn't have this any residue in proximity of the molecule. So this might explain higher potency of compound 3 in 47/7. And also with Dara and Andrea, we did HDX structural proteomics analysis. And we could see here highlighted in color some of the regions that were protected with the compound 3 in USP47 here. and also some regions that were in perturbation with the inhibitor in USP7. So this study confirmed differential binding modes between compound 3 and 47 and compound 3 and 7, indicating binding switching loop rearrangement in USP47 and potential engagement of the activation peptide. So USP47, when becomes activated, has some structural rearrangement around the catalytic site and the ubiquitin bind site called switching loop. And this is important for the activity of USP7 and 47, and involve also a switching loop that maintains this, an activation peptide that maintains the switching loop in the active conformation. So this is the activation peptide in yellow that will bind in this area of USP47 and USP7. And based on the structural studies, we know that compound 3 when it binds to USP47 induces some changes that involve these activation peptides, and these regions are not present in USP7. So this information might explain why compound 3 is more potent and selective for USP47 compared to USP7. And this information can be used to generate either USP47 only or USP7 only specific inhibitors. So, as I mentioned before, one of the things we wanted to use this tool, research tool, is to try to see whether the biology that has been described for some of these deregulating enzymes also can be replicated when we use these small molecule inhibitors. So it's been described that USP47 and 7 are involved in inflammation activation. So this is a process that happens in the context of inflammation, and involves the formation of a cellular structure called inflammasome that eventually leads to the release of interleukin-1 beta and induces a type of cell death called pyroptosis. So when we look at the effects of compound 3 in inflammation activation in a human leukemia monocytic cell line called TSP1, we could see that treatment of this cell line with LPS plus niriesin, these are two drugs that are used by routine to induce inflammation activation. We can see a strong activation. However, when we treat the cells with the inhibitor, we can see a complete inhibition of the inflammation. So these effects of USP47 and 7, they were reported in regulating the ubiquitination of a protein called NLRP3, which is a very important component of the inflammation. And when we treated with increasing concentrations of the USP47/7 inhibitor, compound 3, we could see modified NLRP3, high molecular weight species of NLRP3 consistent with ubiquitination. And we could see inhibition of several markers of inflammation. For example, when you activate inflammation, you can see a cleavage of caspase-1. So caspase-1 is normally migrates in the gel at 50 kilodalton, but then when it's cleaved upon inflammation activation, it runs in the gel at around 20 kilodalton. So we could see inhibition of this cleavage with compound 3. We could see inhibition of the cleavage of pro-IL-1 beta, interleukin-1 beta, which is one of the markers of inflammation activation, and also gasdermin D, another protein, in this case, marker of pyroptosis, that is the cleavage is inhibited by compound 3. So This tells us that compound 3 really phenocopies what USP47 and 7 do in cells in terms of inflammation activation. So OTD7 is another ubiquitinating enzyme that we could see was inhibited by compound 6. And the interesting feature about biology of OTD7B, which is also called CESAN, is that regulates hypoxia-inducible factor HIF1 alpha, ubiquitination, and levels. So when you inhibit OTD7B, HIF1 alpha is ubiquitinated and degraded. So we could recapitulate that. So HIF1 alpha is accumulated in hypoxia, and when we have increasing amount of compound 6, we can see that HIF1 alpha is not able to accumulate in hypoxia anymore and is degraded. One interesting thing is that this is not happening in OTD7 knockout cell lines. We have a colleague here in the institute that is very interested in how hypoxia regulates hypoxia, HIF1 alpha induced in hypoxia inhibits SARS-CoV-2 replication. And we could see that when we have cells in hypoxia, they are refractory to COVID infection, as you can see here. But however, when we treat with compound 6, since we rescue HIF, so we prevent accumulation of HIF1 alpha, SARS-CoV-2 is able to infect the cells. So HIF1 alpha is an antiviral, and with this drug, we decrease the levels of the antiviral, and COVID is able to infect these cells again. And finally, we also tested whether the compound 25, the USP5 inhibitor, has some recapitulates the biological functions of USP5. USP5 is a ubiquitinating enzyme that modulates interferon production by regulating the ubiquitination of IRF3. And this has been described that in a screening method that USP5 is the ubiquitinating enzyme that can prevent, upon inhibition, viral replication. So first thing we did was looking at interferon production in cells treated with USP5 inhibitor, and you can see here that USP5 inhibitor, when we induce interferon production, can increase compared to the MSO control interferon production significantly. And also we could, with the help of Peter, because again, he has this SARS-CoV-2 replication, viral replication models, we could see that increasing concentration of USP5 inhibitor can indeed inhibit viral replication. And we include a positive control, remdesivir, which is a inhibitor that is used in an antiviral that is used in the clinics. We could see that our USP5 inhibitor is 10 times more efficient than the inhibitor in the clinics to prevent SARS-CoV-2 replication. So as a take-home messages, I hope I convince you that DAPs are interesting therapeutic targets in disease, and that we have established methods to study them. So I explained today glide peptidomics as a technique to identify substrates upon inhibition or genetic inhibition of the ubiquitinating enzymes. And I also explained how ABPP, activity-based protein profiling, can be used to profile the ubiquitinating enzymes. So I show you how we optimize ABPP workflows to increase sensitivity and throughput, and how eventually we were able to use this methodology to screen curated small molecule libraries to discover DAP inhibitors. This approach has at least 10 times higher hit rates compared to traditional high throughput screening. And at the same time, we'll inform you about selectivity in cellular context. So we believe it's a very powerful way of identifying DAP inhibitors. And then through this proof of concept study, we expanded the repertoire of electrophilic groups capable of targeting the ubiquitinating enzyme and identified novel USP47/7, OTT7B, and USP5 research tool inhibitors. And we show how activity-based protein profiling can be used to characterize this DAP inhibitor and optimize them. So, for example, you could see in the case of USP5, thanks to activity-based protein profiling, we could see they were not permeable, and this will allow us to generate new derivatives that were more they were active in cellular treatments. And we also use compound 3 in combination with structural ubiquitomics or SDS-MX to study the molecular basis for USP47 and 7 inhibitors. And importantly, all 3 inhibitors confirmed previously reported biological roles of USP47, OTT7B, and USP5. And finally, to thank the lab. So this is my lab, and especially Alice and Simeon were involved in this APPP high throughput drug screening, and also collaboration with Kessel's lab, Scofield lab, which are the chemistry lab here in Oxford, and other collaborations, especially with Peter, with the viral models, Andrew, the structural study, Dara, structural proteomics, and other collaborators. So, and of course, the founders that are supporting my lab. Yeah, happy to take any questions if we have some time. Yeah, we have 5 minutes, I think.
Awesome, thank you. Just feel free to unmute yourself.
Eden, this is Allison at Wesleyan. I'm curious how you guys maybe I missed it, but I'm curious how you guys got such specificity with your probes. Are they like, how did you get that specificity? It looked like it was just a cysteine binding probe. Am I wrong?
Yes, yeah, exactly. It's impressive that let me show you. Go very quickly to that. We don't normally see such selectivity with ubiquity-based probe, but in these cases, we have ubiquitin as part of the probe. Is very selective, and we don't need to use biotin enrichment and very stringent washes. Just with the HA antibody, we can enrich very selectively for the ubiquitinating enzymes. I have volcano plot like this. You just see, yeah, that's super clean.
So the probe is a ubiquitin with a covalent cysteine binding thing, and then the HA tag. Okay.
Yes.
Got it. So that specificity, that is ubiquitin.
That's the key, is to have ubiquitin.
Ubiquitin. Got it.
Yeah, it's very.
I'm like I'm on my iPad. I'm like squinting. I'm a chemist. I'm like squinting at the
molecule. Yeah. Other probes, small molecule. So you might be familiar. They has generated activity-based probe for DAPs based on small molecule inhibitors, and they are very promiscuous. So if you look at one of his probes in a proteomics analysis, you will see a lot of proteins that are not DAPs. So this is something very specific for this particular probe. Yeah, they are very clean and selective. Yeah.
Okay, great. And then I had another question. Do you think that, so we're thinking about doing some IP experiments ourselves to pull USP7 out of cells. And do you think that, do you think you get better IP if you do this with your tag? Like if we did an HA pull down rather than a USP pull down? What do you think?
What is your, what do you want to look at? You want to do interactomics by mass spec, or you want to do COI-PITs, or what?
We want to just look at expression levels of USP7 and the mutants in cell lines. Just wondering how good the USP7 antibodies are versus an antibody for HA, which I know would be really good.
I know the antibodies against endogenous USP7 are good for Western blot. but I haven't done any USB7 IPs. So
Yeah.
you need to try that first. I
Yeah.
mean, if it's to look at the expression, I will go always for the, endogenous protein. You have the different mutants, I will use endogenous protein, unless you are overexpressing them. If you are
No.
overexpressing them,
Yes, they're
then
in.
you're stuck. Yeah,
Okay, thank you.
yeah, no worries.
Really nice work. This is cool.
Thank you.
Yeah, very impressive. Can you expand a little bit on using these probes for assessing activity of USB7 mutants?
Yes, so this probe, they have a strong affinity for the catalytic system. So in case you have mutant, they will alter the reactivity of this residue quite dramatically. You will see changes with these probes. If not, it might be too strong binding, and it will give you equally reactivity even with the different mutants. One way of going around that will be to make warheads that are less reactive, maybe like the alkyne warhead that might be a bit less reactive. You can also play with incubation time. You normally do 45 minutes, 30 minutes, but maybe you can start with shorter time points, and that might allow you to detect these changes in activity. We had to do that with non-covalent reversible inhibitors because, yeah, they will compete with the probe, but only if you don't incubate long with the probe. If you incubate long with the probe, you won't see any inhibition. So you need to play with the incubation times. Yeah. I think they could
Would you
work.
need, sorry?
I think they could work to look at the different mutants.
So will you need cell lysates or cells with endogenous USB7 mutant expression in there, or can you use purified proteins, or can you use just the catalytic domains purified?
I mean, since this probe works so well in cellular extracts, I will always look at the endogenous protein because it's more relevant always. And this allows us to look at the endogenous protein. And USB7 is one of those that is labels very well with the probe, and you are able to separate the unlabeled versus labeled very well. So for example, you have USB24, which is 300 kilodalton. You barely see the shift, right? But USB7, you can see very nicely the labeling and also the shift. So this will allow us to, by Western blot, just labeling lysates will allow us to look at endogenous differences. Yeah. So yeah, just cell pellets that then we have a specific way of lysing them without detergent and with glass beads, and that will be enough. Yeah.
So if we sent you, can we send you material to test?
Yeah, I mean, yeah, it will be great. Happy to help with that. Yeah.
Thanks.
Yeah, I have several people in the lab that they are able to do it now, so we can spread a bit the experiments. Yeah.
Great, thanks. Can I ask another question?
Of course. Yeah, yeah.
Okay. Your USB47 work was really interesting. The structures that you showed, was this activation, was this C-terminal peptide? Is that a crystal structure? Is that AlphaFold modeling?
This is modeling. Yeah, there's no crystal structure for human 47. Andrew did all this. It's from for another organism, which I don't remember now. There is one structure from that.
Yeah,
And then using, I guess, some of your structures and our structures with the forma compound, he also was able to model the catalytic domains, and binding to the compound.
Mm.
Yeah.
Mm.
Yeah. So yeah, he came up with this activation peptide because I will never come up with that, and he realized that this is where the activation peptide lays after activation, upon activation. And he saw that these residues were the ones that they were affected by compound binding 47. And he came up with this explanation to explain selectivity, in addition to this arginine that USP47 has and USP7 doesn't have in that position. Yeah.
Thank you.
Yeah, no worries. Okay, so I guess I'll stop sharing.
That was really great. And I will CC you on our wrap up email so everyone has your contact information.
Excellent.
Thank you so much for your presentation today.
No, thanks for inviting me. Yeah, it was great. Thank you.
Have a great day, everybody.
Thank you.
You too.
Bye.
Bye.
Thank you.